Okay, so we're going to start here with the central figure of our standardized, this is part 7, page 87, our standardized cell right here. And there's a number of organelles that I'm going to get to, but first I'm going to begin with the central organelle right here. I'm also going to point out that cells have quite a few different morphological formats, and I think I've already mentioned that there are cells that are like muscle cells that are inches long, and individual neurons that are feet long, going from the tip of the toe to the spinal cord. One single cell that is microscopic in nature. You can't see it, but it travels a great gross anatomy distance. So different shapes, but nevertheless we all have a nucleus in most of our cells. There's exceptions like the red blood cell, but generally speaking, cells have the instructions. And we go from an individual zygote, a fertilized egg, with the original 46 chromosomes of instructions, and they're enclosed in what we call a nucleus here. The nucleus is a double membrane unit. It's got a little cistern right here, the space between the two layers of membrane. We have pores to let information in and out. This is the reserve book room, in my opinion. This is where we keep all of the original instructions as to who we are and what we are. We actually have the history of our phylogeny, and through embryological development, it's observed that we go from an individual cell to like a little flatworm to a something like a fish with gills, and then something like a little piglet with a tail, and then finally we look rather human by the end of the first trimester. In particular, we have a short little human in there, slightly large in the head, but nevertheless elongating in the body and looking fairly human. Okay, so and all of the cells, all of the trillion cells that will be there by the end of gestation at the time of birth, they're going to each have a set of instructions. So a set of encyclopedias, some of which will be pulled to the side, condensed, and not being used, and then a lot of it also on. That is to say, there will be some naked DNA, and there will be sequences of actual genes. That is to say, there's going to be sequences of instructions. So the book will be open, so to speak, at certain pages, and we'll be able to use those instructions because they're available. And so this cell will be what this cell is. It'll be an epithelial cell in the skin. It will be a muscle cell, a neuron. It will be whatever it is that the instructions have differentiated to make available. Okay, and there can be no DNA leaving the cell. The cell can replicate and can redistribute its DNA evenly with dosage regulation, and that's what mitosis is for. But meanwhile, we need to have the instructions remain in the cell. There are bacterial cells in which the instructions are actually in the cytoplasm, sort of, how shall I say, moving about the cytoplasm, whereas here we don't have that situation. We actually have a storage unit, and there's a very discrete in and out here in a little pathways in which information can be, how shall I say, brought out here. And the purpose of the information is to make protein or polypeptides, so sequences of amino acids. And that's what our so-called phenotype is all about. So if we become humans with various characteristics representing gender or representing a certain height or other types of indicative capabilities, usually family-based, the size of our nose, the color of our eyes, there's a number of features that are biologically based that you can more or less count on for being there. There are certain immunological properties and genetic properties that can be found in terms of the genetics of our little name tags. We'll talk about that at some point. Okay, so how do we deal with this information if we can't leave the reserve book room. Well, like any reserve book room, we can make copies, and then we can leave with the copy. So we look things up, we make a copy, we leave with the copy. It's a little complicated here because we're going to be using 3 types of RNA. And there's the type of RNA in which there's a machine that's made out here. You can actually see some of these machines if you look very, very closely. Let's see if we can get close enough here. Yes, you can. You can see what's called rough ER. And there's pictures in the book here that we can look at, and we will, to see that there's little machines that can take a sequence of a gene. And how do we get that sequence out to them is that we make an elongated copy called messenger RNA that leaves the pore right there. Another thing that happens is that we have stored here the parts for that machine out here called the ribosome, and we can bring those parts out and arrange them to make the machine that actually copies the sequence of messenger RNA and produces the sequence of polypeptides. When we copy the DNA to RNA, we call it transcription, and when we copy the transcript then to make a sequence of amino acid peptide bonds so that we have the primary structure of a polypeptide or a protein, we refer to that as translation. So transcription, make a copy. Translation, we translate that copy into a sequence of a completely different chemical nature, from nucleic acid to protein. Okay, and we have a third type of RNA, which actually little tiny palindromic RNA that forms the shape of a T, and it goes out and it actually finds amino acids and binds to the pool of free amino acids in the cytoplasm. So there's amino acids out there from nourishment that comes with your daily diet. Okay, and so the tRNA can bind to that and bring those amino acids over to the machines that have been made by the ribosomal RNA. Okay, so we'll take a look at that. Meanwhile, the pathway out of here is going to lead us all around, and I'm going to consider the organelles basically in the sequence in which they appear. Okay, I hope you're okay with that. So I'm just turning now to the page where we're looking at that outflow that I just described to you. So here we have that outflow, and we have here basically we're making a copy right here. We're transcribing a length of DNA, a gene's worth of DNA, shall we say, into a length of messenger RNA. The messenger RNA is going to exit. Here we have the exiting messenger RNA. It's going to come down through here, and then it's going to be, it's hard to get backed up enough to look at this book, but basically we have here a ribosome. So that's what's positioned right here. Let me come back far enough to look at that properly. There we go. Okay, so we have a ribosome here. There's it looks a little bit like a catcher's mitt, and it can sort of close its palm down on this messenger RNA as it comes through. And what it's doing is it's reading three base pairs at a time. It's lining up the base pairs with a particular amino acid. The amino acid positions itself, and then it basically meets up with a second tRNA. The second tRNA over here is lining up and delivering its amino acid to the ever-growing sequence, and we have a sequence of amino acids growing off there. So that's what you need to know, is that we're producing sequences, and ultimately here we have sequences that are being made. Here is a single messenger RNA, and it's actually being drawn through several different ribosomes, and this one has seen most of the message. I guess it's going this way, and as a result of that, its amino acid sequence is longer. This one is going along, this one's going along, this one's going along, and so the deal here is that we're actually this is rough ER, and it's rough because it had these things on the surface, and we're producing the protein inside the endoplasmic reticulum. So we'll get to that again.